you can start.
Okay, thank you. So, thank you all who are connected for your interest in this presentation, which is a presentation on how to essentially approach a biological problem using screenings, and in particular screenings based on multiparametric microscopy, so high content microscopy. And the presentation will not be technical because I think that it is more interesting that it is more conceptual on the kind of approach, and basically a vast part of it will be just showing a few of the problems that we tackled by screenings and how we adapted our research to that. I mean, if this can be somehow inspirational for other kind of approaches some of you might undertake. So basically, I will speak briefly about why screenings are interesting, then what we can do here and what are the principles of high-throughput screenings and high-content microscopy. And then I will give some of the examples as examples of basically approach more than showing scientific results at the end. I think that approaching a problem with the screening is really conceptual, and perhaps some of you could already have seen these slides, but I like them very much. And the more I think of them, the more I think that really they describe how research is usually carried out, and basically for those who are not young anymore, as I am, I remember that in the days of my PhD and then several the decades after that, there was a sort of mantra that research should be hypothesis driven. So basically you observe something, you make an hypothesis on what could be the mechanism, and then you perturb the system and prove whether the mechanism is correct or not. And this is what I call the biochemist approach. So the approach that led to an enormous amount of discoveries in which basically you center your attention on one gene, one protein, one mechanism, one enzyme, and then you perturb it by mutating it, by changing its localization, by studying the protein that binds to the one you are interested, by mutating if it is a gene, so on and so forth. And this is very interesting, and this has provided an enormous information in biology. However, the limitation is that you never know whether it is relevant or not. And even if it is relevant, you never know whether there is something that is more relevant of that. So basically, you cannot prioritize whether the kinase you are studying is more important than any other kinase in the cell, or if the gene you are studying has an effect that is higher than any other gene in the same family. Then, when the omics world was rendered possible, first by DNA sequencing for RNA-seq, for example, and then by proteomics, lipidomics, metabolomics, interactomics, you can read the omics here on the slide. Then the research changed again. And here, basically, all these omics techniques have permitted for the first time to provide scientists a sort of broad and comprehensive view of what's going on. I call it the snapshooter approach, or the photographer approach. So basically, you take a picture of what's going on, then you perturb the system, and you take another picture, and you see what are the changes. Typically, you have your cells, you do RNA-seq profile, then you add the growth factor, you see which are the genes that have changed their expression levels. fantastically interesting, but then you end up in lists of modifications that if you are a good bioinformatics, you can run through the software that can group them in gene ontology, in pathway analysis, and so on and so forth. If you're not a good bioinformatics, you have a list of modification in Excel files, and at the end of the story, if you look at your Excel files, then you pick up genes that might be interesting when you read, for example, a paper in the literature and you see whether they apply to your system. Or if you are good bioinformatics and you have pathway analysis and gene ontology and gene categories, then this is very interesting from the observation point of view, but at the end of the story, it's very difficult then to pick up what could be the master gene. So if you have a pathway that is modified, then very difficult to understand which within that pathway is the gene that is crucial to induce the phenotype that you observe. And if you think, however, in the history of biology, some of the big discoveries have been discoveries in which people have not tested proteins or genes, candidate proteins or genes first, and didn't have omics available, but they did screening. So basically, the search for function without knowing what was the responsible factor that was inducing that function. This is one of my favorite experiments in the early 80s when oncogenes were discovered. And so several laboratories, Stuart Aronson at the NIH, Mariano Barbacid, they took cDNAs from cancer cells and transfected these cDNAs in NIH-33 fibroblast, which is a kind of fibroblasts that grow attached to plates, and they are in which cells are inhibited in proliferation when they are confluent, so they have contact inhibition. And basically, if a cDNA for an activated oncogene enters one of these cells, they lose contact inhibition, and the cells start growing one on top of the other, they form foci. This is one of the way that oncogenes were discovered. Or in another example that I have seen in my career, in my previous interest, I've been working with HIV for almost 20 years. And I remember that when the virus was cloned and sequenced in 1983, and it was immediately clear that the receptor for the virus was the CD4 receptor on lymphocytes and macrophages, simply because it was a good hint that patients progressing towards AIDS have a progressive diminution of the number of CD4 positive T lymphocytes. So people immediately took cells that don't express the CD4 receptors, like HeLa cells, transfected the CD4 cDNA, and then tried to infect these cells with the virus, and the virus didn't enter. It was not internalized. And so it was immediately clear already in the mid-80s that there was an additional receptor that nobody could find until, I think, 17 years later, a laboratory at the NIH, Ed Berger, was the PI, did a screening experiment. So he took a cDNA library again from CD4 positive T lymphocytes, believing that these are infected, so they must have this co-receptor, transfected HeLa CD4 cells, and then he selected cells that at that point were infectable by fluorescent HIV. In this way, the chemokine receptors, one of the chemokine receptors, CXCR4, was discovered for HIV. Going more towards our days, Yamanaka in 2006 published his famous Cell paper in which he described for the first time the discovery of 4 factors that induce differentiation of fibroblasts into embryonic stem cells by taking a collection of lentiviral vectors and testing various combinations of these in a blinded manner, which eventually, from which eventually he found the 4 factors that permit formation of iPSC cells. So if you want to conceptualize this, if you do expression analysis, you ask yourself whether there are messenger RNAs, microRNAs, link RNAs that are present in certain conditions and not in another conditions. While if you do screenings, you can screen the same factors, so messenger RNA, microRNAs, link RNAs, or proteins, but for the function they induce. So you don't observe what are the changes, but you search for those that induce these changes, with one essential additional difference, that if you do observational analysis, you don't know what is important or not. You try to guess it before, because an mRNA that changes 10 times probably is more important than an mRNA that changes 2 times. But this is just an assumption. Well, if you do a screening and you search for a phenotype, then you can rank the potency by which any of the factors you have tested change that phenotype. Again, to conceptualize, there are 2 types of screenings that one can think of. One is screening with a pooled library. So it means that, for example, you have a collection of vectors with shRNAs, or single guide RNAs for CRISPR, or cDNAs, each one in a vector, in a plasmid, and all mixed together. So you have a collection of thousands or tens of thousands different vectors or plasmids, and then you put these into cells and search for the phenotype. So you screen for the phenotype you want. But at this point, the phenotype, if it is a proliferation, you can simply wait. The other cells die, and the one that proliferates has a selective advantage. Or you can treat the cells with a drug and screen for a factor that renders these cells more prone to survive. Or if you don't have a selective advantage by the factor, the only way you can screen is for a phenotypic marker, for example, through flow cytometry, and then you sequence what was in the cells that you have selected. Much tricky, very tricky. Instead, another possibility is to do arrayed screenings, in which you have all the molecules in your library separated from all the others. So, for example, in vials or in plates, so you have collection of SIRNAs, LNAs, single guide RNAs, small molecules, cDNAs, but each one in a different tube or well in a plate. And then you screen these individually. Obviously, if the libraries are big, then you have to screen them robotically. So you have to have a robotic machine that dispenses and do all the kind of assays. And then you have 2 ways of reading the outcome. One is by lysing the cells and then measuring absorbance, luminescence, fluorescence, according to a reporter gene you have in the system. Or another powerful one is the possibility to have systems of automated microscopy that simply take pictures of the different wells, and then of a software that analyzes these pictures, and which is called high content analysis. These are some examples of high content analysis. Simply it's taking a picture of what's going on in the cells, like in a microscope, and then having a software that transforms a microscopic image into a computer-generated image. For example, you have a reporter protein that stays in the cytoplasm and goes to the nucleus, and that is responsive to a promoter that gets activated. The software then picks up the cells and transform this into a digital image. And the kind you can screen depends on the software. So basically, the microscope simply takes images in an automated manner. And then it is a software that somehow transforms these images in images that can be analyzed quantitatively. These are, for example, some examples. We speak about cell proliferation later because it is one example that I will show you more in detail. But for example, this is viral infection. This is a virus, it's a rotavirus that forms replication factories in the cytoplasm. And basically, the software digitalizes the images, recognizes where the nuclei are, and each of the dots can be quantified and identified. Or cells that are damaged express gamma-2AX in the nucleus, and then the software can recognize the nuclei and then understand which are the ones that have gamma-2AX. This is an example with bacterial infections. So bacteria that are fluorescent, the software here is an easy task, you can simply measure the fluorescence inside the cell, but provides you a quantitative image and a quantitative information based on images. This is, for example, a more sophisticated application in which there is a wound in a cell monolayer, and then this wound is repaired by cell migration, and you can have quantitative information on the speed and the rate and efficiency at which this wound is repaired. The system is a system you have here. You have seen this quite a few times in my presentation. I'm showing it in more technical details. So basically, this is a system in which there is a robot can do all the dispensing, an arm that slides across the instruments and can deliver the plates to a standard cell incubator adapted obviously for this automated use, and then through this robotic arm from the robot that dispenses the treatments and put the cells in the incubator, the cells can be retrieved from the incubator and processed through a liquid handler for absorbance, luminescence, and fluorescence, or fed into a high content microscope, which is this operator CLS that does the quantitation, that does the images. Then the analysis of the images can be also performed offline by a software that is connected to the system. What is interesting is obviously which libraries one has available, and what we have here at the school is a whole genome siRNA library, so approximately 18,000 siRNAs. Each siRNA is composed by is a mixture of 4 different siRNAs, so it's a pool of 4 siRNAs, each one at a lower concentration, and this is a way by each targeting different regions of the same gene, the same mRNA, which is a good way. It is a commercial from Dharma, it's a good way of trying to avoid side effects. Then we have a whole genome library for mouse, and we have a library of microRNA mimics from for mouse and one for humans. We have a library of antisense oligonucleotides with LNA modification to increase stability against microRNAs for human and mouse. And we have 2 collections of FDA EMA-approved drugs, which are one of approximately 2,500 drugs and the other 1,200 drugs. Then we have 1,200 plasmids encoding proteins from the mouse secretome. The mouse secretome has approximately 2,000. Here there are 1,200. And then we have 500 plasmids encoding single guide RNAs against the vast majority of receptors expressed on the surface of cardiomyocyte. These are guide RNAs that can be used in CRISPR-Cas9 screenings. A few examples of what we did in terms of screening in the past to give you an idea of what can we screen and how interesting is this fishing expedition, where you know that you induce a function, you don't know what causes that function, and then you start studying what comes out and understand what is the mechanism. So this is work we did a few years ago, trying to answer an interesting question. That is, you know that AAV vectors are fantastic tools to transfer gene in the heart, in cardiomyocytes, and they are fantastic tools to transfer genes also in other tissues, like retinal cells, where they go to neurons, and also to retinal pigment epithelium cells and photoreceptor, to brain, especially in neurons, skeletal muscle fibers, and then cardiomyocytes. The question here is, why don't they work in fibroblasts? Why don't they work in endothelial cells? Why don't they work in all cells that grow in an incubator in the laboratory? So why they are restricted to post-mitotic cells? And there is very little known about cell permissivity to AAV vectors. So we decided to tackle this with the screening. And the screening here was to have an AAV vector with a constitutive promoter, the CMV promoter, driving expression of the luciferase gene. We proved that the vector is working. And then the assay was to have a library of SIRNAs targeting the human genome, arrayed on 384 well plates, which is what we have here now, thanks to Luca and Eduardo, just in these days are preparing these libraries. And then basically the SIRNAs are plated by the robot in the plates. So we do reverse transfection, so basically together with lipids. And so basically we have the plates with lipids and the SIRNAs. And then we add on top of this, in this case, we added HeLa cells. And then infected after 48 hours, so we left the SIRNA downregulating each respective target for 48 hours. Then with the robot, we dispensed the A-field luciferase, waited additional hours, and then lysed everything and tested for the expression of firefly luciferase by the liquid handler. This is a way how a screening looks like. And so basically each of these dots here is one of the 18,000 genes analyzed, where simply we express the luciferase activity versus cell viability. The advantage of high content microscopy is that you can count nuclei, for example, and see how many cells you have in your well. And so you immediately understand whether your the reduction you could have, for example, in luciferase is artificially due to the reduction in cell viability. And so you discard what is below a certain threshold of viability. And basically, if you plot this, you find that the vast majority of genes stay in a range that's not statistically significant, but there are, in this case, really a very large number of genes that were statistically significant. We were surprised at the number of genes, but then we understood that it is a matter of AAV biology. This was a discovery that we made, because what we found is that any time you have a gene that increases DNA damage to the cells, so that touches the DNA damage response, basically you increase in AAV permissivity. Just to give you an idea, all these genes that increase AAV permissivity are all also genes that induce gamma-2AX foci inside the cells if you knock them down. I'm showing this because this is another way of approaching. We screened for 18,000 genes. We found a series of genes that we believe have a phenotype similar, in this case, induction of DNA damage. So you can go back to your library and pick up, so cherry pick the genes you are interested in, and do a secondary screening to search for a certain phenotype. For example, these are all of the 1,500 genes that we tested for this time induction of gamma-2AX foci. This led us to a model by which basically AAV, when it enters the cells, it is recognized by the DNA damage response proteins and it gets inactivated. So and these proteins are very active in all replicating cells, but they disappear from cardiomyocytes because the cells exit this cycle. Speaking about, again, DNA repair, you know that, and this is work by Eduardo here, you know that cardiomyocytes are post-mitotic cells, and everybody knows that this is the main problem by which these cells don't replicate and the heart doesn't regenerate after damage. But there is another consequence to this, that is that if you induce a double-strand break, for example, you express the Cas9 protein and a guide, this break in replicating cells can be repaired through pathways, non-homologous enjoining, in which the double-strand break is recognized by the Q protein and the DNA PKCS and other proteins, and then there is error-prone repair with the insertion of mutations or small mutations, small deletions, which is very frequent. However, replicating cells can also repair DNA damage through homologous recombination, by which there is a complex of proteins that's called mRNA, MR11 or 50 MBS1, that recognizes the damage, reduces one of the extremities of the damage to single-stranded DNA, and this single-stranded DNA, together with the RAS51 protein, invades the other chromosome to have homologous recombination. If instead of the other chromosomes you provide the cells a large amount of the same template, then homologous recombination can occur with this template. You have perfect correction. The problem of cardiomyocytes is that they exit the cell cycle around birth, and so the MR11, RAF50, and BS1, who are very expressed at high levels until the first week of life, suddenly they disappear. And so basically homologous recombination does not occur. And so what Eduardo wanted to do is to find the system, to find if there are microRNAs that can induce homologous recombination in cardiomyocytes. So there he had to invent an assay. So high-throughput screening are based on assays. So the most robust is your assays. The easiest is that you will find the phenotype, a functional. If you have a strong functional phenotype, the easiest will be that you find a molecule that functionally modify this phenotype. So here he constructed an assay in which basically he delivers to cell a green fluorescent protein that is mutated. So basically it is black, it is inactivated. A template which can correct this mutation through homologous recombination, so render the green fluorescent protein again green fluorescent, and then he provides Cas9 and the guide cleaving in proximity of the mutation. He transfects this construct into cells, and then he analyzes the efficiency of transfection by an antibody against Cas9, against the GFP that recognizes both the unfolded, so the mutated and the folded protein, and then by fluorescence or an antibody against GFP, by fluorescence that measure the active protein. So basically he has an antibody against GFP in red and fluorescent protein in green. And this is how the screen looks like. This is the screen was for microRNAs, and this is a control microRNAs, and this is a microRNA that was positive. So basically, efficiency of transfection is identical in all conditions, but here you also see many more green cells, which is the product of homologous recombination. In these screens, in this screen, for example, we screened 2,000, a bit more than 2,000 microRNAs. We found only 21 that were effective, and it was very interesting to see that 10 of these had exactly the same seed sequence. And this is a very strong indication in screening that we are hitting something that is really important, because having the same seed sequence means that they all worked through a common mechanism. And now, I mean, after the screening, Eduardo is trying to see whether this works in cardiomyocytes, and it does. So these are the first cardiomyocytes in which he achieved the molecules recombination by delivering these different microRNAs. Speaking of cardiomyocytes, another very successful screening, there's been one, perhaps the first one that we did several years ago in Italy, was for microRNAs that induce cardiomyocyte proliferation. We like very much microRNAs for these screenings because we are screening for phenotypes, and sometimes phenotypes are difficult to achieve. So think at forcing a cell to have homologous recombination, or forcing a cardiomyocyte to proliferate. You need to regulate very complex systems inside the cells, and microRNA are perfect for that because each microRNA targets tens of hundreds of different genes. So they have been evolved by nature to do complex tasks as those that we are speaking of. So it's not a matter of activating a kinase, but it is a matter of changing completely cell behavior. For example, a cardiomyocyte that has to proliferate has to downregulate or disassemble all the sarcomeric apparatus, has to reactivate expression of all the cell cycle genes, and has to couple then cytokinesis with the presence of this enormous cytoplasm with a disassembled sarcomeric structure. So very complex phenotypes that microRNA targeting multiple genes are more easy to impact on. And then there are many microRNAs, so more than 2,000, but not as many as other categories. So doing a screening for 18,000 genes can take a week just for the screening, and it costs an enormous amount of money. So an estimate cost here, not including the people doing the screening, is around £1 per well. So if you start doing a screening of the human genome in duplicate, you already, just for the screening, you invest £50,000, just to give you an idea. MicroRNAs are much more approachable in this respect. So the idea here was to find there are microRNAs that induce cardiomyocyte proliferation. The screening here was a screening in which we reverse transfected microRNA on 96 well plates, and so lipids by microRNAs. Then we took primary cardiomyocytes from rats, plated on top of the microRNAs, and then added an analog of thymidine, which can be either BRDU or EDU, that is incorporated into cells when the cells duplicate their DNA. So the cell incorporates this instead of thymidine. And so what happens is that cardiomyocytes that incorporate EDU or BRDU, then the EDU can be seen by chemical technology called click chemistry, BRDU with an antibody, but bottom line is that the nuclei of cells that replicate become can be visualized versus all the other cells. This is a result of the screenings. The screening was performed in rat, then repeated in mouse, and this is the overlap between the two. I'm wanting to show you here how the software works in this case. So these are primary images of a control cardiomyocyte and cardiomyocyte with one of the pro-proliferative microRNAs. So the cardiomyocytes here are in green, the EDU is in red, and the nuclei with DAPI in blue. And then the software picks up this image, makes the cell contour, and then you can instruct the software that anytime there is a red nucleus, but this nucleus has to be within the cardiomyocyte and not outside the cardiomyocyte. Nuclei outside cardiomyocyte are fibroblasts. Then count it and give it a yellow color so I can visualize it. And so basically at the end, one counts only replicating nucleus inside the cardiomyocyte. In normal conditions are about 10%, and this is the effect of one of the most successful cardiomyocytes, which rise this percentage to almost 50%. Now, obviously, this is very interesting, but we have a microRNA that drives proliferation. But the obvious question is, what are the target genes of these microRNAs? This is the obvious question that any reviewer or any scientist poses to himself. And again, a powerful way of addressing this question is through screenings. This is what we did in the case. So basically, we wanted to understand how these two microRNAs work in driving proliferation. So what we did was to transfect these microRNAs into cardiomyocytes. And then we did next generation sequencing. And we had the list we were speaking before. So basically, we have a list of genes that are downregulated and genes that are upregulated. So if you took them cumulatively, we had 697 genes that are downregulated and 1,000 that were upregulated. And here, obviously, you can do pathway analysis and gene ontology, so on and so forth. Instead, if you approach this through screening, this is what you can do. So basically, there are 696 mRNA that are regulated. Well, let's take the SIRNAs against each of these and test. Sorry, it is here. I don't have the highlights. And test for each of them if individually they can stimulate proliferation. So basically, what we did was to take 600 genes that are downregulated by these microRNA. We cherry-picked from the library these 600 SRNAs, and then knocked them down individually, and we found, here it is, sorry. And then at the end, we found that, for example, all these genes which are downregulated in the presence of the microRNAs, also by themselves individually, they can stimulate cardiomyocyte proliferation. So we have a functional proof that the activity of these microRNAs goes through the downregulation of some of these genes. So again, this is a functional way of approaching the problem. By the way, there were some of these that were particularly interesting because these are genes that target the cytoskeleton. And so we went on this story and found that there are some pathways that are common for these proliferative microRNAs. One of these pathways is a pathway by which there is an impact on the actin cytoskeleton. So in the cells that we treat with these microRNAs, they become roundish and they start expressing actin at the cortex of the cells, which is called cortical actin or stress fibers. You see the difference between a control and one of these microRNAs. This effect is not specific for microRNAs. We can see even in fibroblasts. These are normal fibroblasts. This is F-actin in green and tubulin in red. If you treat this with this microRNA, you see that there is formation of these stress fibers, which was a hint that the cytoskeleton was involved. Well, let's approach this through systematically. We cherry-picked 400 SRNAs against all known cytoskeletal proteins and all known sarcomeric proteins, and individually downregulated each of them. And we found a series that were very active in driving cardiomyocyte proliferation. This is the kind of approach we like, an approach in which at the end we have a screening for function. Another type of screening that is relatively easy is a screening for protein stability. Here, the setting is HIV infections, and you know that HIV enters the cells, and then in the cytoplasm there is reverse transcription of the cDNA, of the viral RNA into cDNA, that this enters the nucleus through a nuclear pore, and then there is an enzyme called integrase that integrates HIV DNA into the host cell chromosome. And it is known, has been known for 30 years, that if you infect cells that don't replicate, so lymphocytes that are resting, then there is no integration. This doesn't work. And what we discovered that the reason why it doesn't work, it is because the integrase enzyme gets rapidly ubiquitinated and degraded. And this can be seen also experimentally. So, for example, if you transfect integrase, you have this amount, but if you wait 1 hour or 2 hours, then progressively this goes down. If you give an inhibitor of the proteasome, MG132, then this is stabilized and remains high. If you remember, the ubiquitin proteasome works because there is one enzyme called the NE1 that interacts with several E2s to transfer a chain of ubiquitin on a target that is identified because it interacts with one of several hundreds of E3 proteins. And this is what is called the ubiquitin conjugation system. And so what we did was, the question here was, which is the E3 ligase that is involved in tagging integrase for degradation? So what Hashim did a few years ago was to fuse integrase with GFP. He transfected it into cells. You see that there are a few positive cells. Treated these cells with MG132 and stabilizing the protein. You see there are many more cells. So and seeing that indeed the integrase is rapidly degraded. However, in the presence of proteasome inhibitor, is stabilized. And so the screening was very straightforward. So what we did was to cherry pick from the library a set of 600 SIRNAs against genes in the ubiquitin proteasome pathway, so all 3 ligases, all the proteasome factors, and all the other enzymes involved, known to be involved. And then these SRNAs were reverse transfected in 96 well plates, and then HeLa cells were plated and transfected with this GFP fused integrase. And then the screening was for siRNAs that render fluorescent integrase more present than in wild type conditions. And again, what we found was a factor that's called TRIM33 that then we studied and discovered that it is a potent stabilizer of integrase. So it is a potent degrader of integrase. So if you knock it down, basically you stabilize integrase for its activity. And the final screening is a screening that we performed here during the lockdown. And here the screening is for SARS-CoV-2. And basically we started from this observation here. And this on the left side, there are normal viro cells in which the cell boundary is stained with gamma glutamine in red, nuclear in white. And these are the same cells after 12 hours, an overnight expression of the spike protein of the coronavirus that induces formation of this enormous syncytia. Here, spike is stained in green. And this is these syncytia are also seen in the lungs of people who died of COVID-19. You see this big syncytia in the lung. So we decided to set up a screening of FDA EMA-approved drugs that block this syncytia. FDA EMA-approved drugs because these can be repurposed against the COVID-19. And here the screening were two that we set up. In one screening, what we did was to load a cell expressing this spike protein with quantum dots emitting at in green, 525, and then mix these cells with cells expressing the receptor for AC2 and loaded with quantum dots emitting at 800. And then basically the idea is that if these cells fuse, the fused cells should have both more than one nucleus and both red and green dots inside the cytoplasm. This is how it looks like. And so basically these are just mixing red and green cells without spike around. And you see that you mix them, but that cells remain either green or red. However, if there is spike around, the cells fuse And then you have these cells that have more than one nucleus and both red and green dots in the cytoplasm. These are some other examples of these syncytia induced by the spike protein. And here the screening is, this is an image from the microscope. Then you instruct the software, and the first thing that you do, you tell the software is trace the contour of these cells, and then find the Q dots in the cells, find the other Q dots in the cells, overlap the two images, and you identify the fused cells. And eventually, so we screened the library, found a few drugs that inhibit significantly formation of syncytia. The other screening that we did with the same purpose was to directly transfect the spike protein inside the cells, and then simply see if there are drugs that block the fusion. Here, basically, the first step in the software was to identify the nuclei and identify the cytoplasm of the cell. And then within the nuclei identified, search for those that are clustered, because the nuclei of syncytia stay one close to the other. And then these are defined as syncytia. So if there are more than 5 nuclei in a single cytoplasm, the software is instructed to call it a syncytium. And in this manner, you can simply quantify the syncytia and so have an idea of the activity of the drug at inhibiting formation of this syncytia. This is how the screening looked like. These are some of the drugs we selected. For example, these are big syncytia that are formed in the control, and this is one of the effective drugs. You see that the cells are still transfected with spike, this is here in green, but they remain very small. And this is a top drug, and one of the top drugs in our screening, big syncytia in the control. With the drug, the cells still express spike, but they remain very, very small. You can see also here. And this is the kind of screening that we do in the cardiovascular field. We are screening also FDA EMA-approved drugs. So we have screened in the past for inhibitors of cardiac fibroblast activation. This was a screening for TGF-beta induced smooth muscle actin activation, so transition to myofibroblasts, and how the drugs can inhibit that. And then we have a collaboration with a group in Lausanne to screen for anti-link RNAs for reprogramming cardiomyocytes. Here's the screening. The assay is the readout is expression of cardiomyocyte specific markers. We have a screening with Andy Baker here at the University of Edinburgh for microRNAs that control proliferation of smooth muscle cells with a target in primary hypertension. But I don't want to go much into this detail of the list, but just to say that there are many applications in which this kind of approach can be taken. I'm finishing up. I think my time is gone, but just with a notion that is that if one decides to do a screening, usually it takes 3 months to set up an assay, 3 days to perform the screening, and 3 years, or well, I'm a bit pessimistic, let's say 2 years to understand how the hits that one has found are working. So what are the molecular mechanisms of action? You will say, well, but 2 years is also a long time. But yes, but it is a long time, but you spend this long time working on a factor, so a microRNA, a cDNA, in a SIRNA, that you know that it is the most active factor for the phenotype you have screened for. So you work these 2 years knowing that you understand the mechanism of action on something that is that has top relevance in your subject matter, so in for the reason we have performed the screening. So you work in a completely different state of mind than working 2 years on a factor that eventually you don't know how powerful it is in vivo or in reality. And so I think that is also much more reassuring in terms that you're doing something that is possibly more relevant. So thank you very much for your attention. I stop here. I'm very, very pleased to take questions if you have any.
If you have any question, either raise your hand or write the question in the chat.
Alex.
Thanks, Mauro. That was a really great talk. I just wanted to ask you a couple of practical questions. So being a leukocyte biologist, I'm interested in like the immediate early responses of immune cells. And I'm thinking about in particular neutrophils, which, you know, if you touch them, they go boom. So one way in which I was wondering the high throughput screening setup might be able to operate in favor of looking at early immediate responses of, say, neutrophils would be in that schematic where you have the incubator. Is there any scope for cooling the plate down so that you can do some kind of a temperature block release type of experiment? So you lower the temperature to decrease the biological activity, and then you add whatever you need to do into the wells and then put it into 37 so that, you know, you kick off the effect of whatever you've added in unison in all of the 384 wells. Is that at all possible?
The short answer is I'm not sure how to answer. The chamber we have is thermostated, but I'm not sure you can cool it down to a temperature that wouldn't allow neutrophil activation. Well, we can check this with Luca later.
Okay, thank you. The other question I had was about the downtime in imaging. So say from well number 1 to well number 384, if this microscope was to take an image across all wells, and let's just say for it's just taking a single channel, I'm wondering what the downtime is from well 1 to well 384.
That depends on how you program the microscope, because you can subdivide the field of each well and take subsequent images of all the subdivisions, so fill up all what you have in the well, or you can take subfields, and then you can take images different resolution and confocal or non-confocal. And so if you have to screen 18,000 SIRNAs, you also have to deal with the fact that basically you want that the conditions are the same from the beginning to the end. And so probably in that case, one wants to screen at a bit lower resolution and a bit a surface that is a bit less extended, and so this can vary. So can vary between few minutes, several tens of minutes. So it depends a bit on the application.
Right. Yeah. Sure.
All this can be calculated in advance, so that the machine basically you set your parameters and tells you immediately, and how long does it take to scan the images the wells, exact wells you have chosen to scan. And then you can change your parameters online and find a setting that is suitable for your experiment.
Thanks. You told us just one more question. You told us how much roughly it would cost for a single well of a 384 well dish for siRNA, but not for microRNA. And I'm wondering how much that would, what the differential is between those two
there is
approaches.
no differential between siRNAs or microRNAs, and we are calculating the costs now in a more exact manner with Steve Bunting, and there will be a different cost for internal and external, but in the meantime, I mean, a generous cost in the sense that the cost just for covering Basically, the reagents is 1 pound per well.
Mm-hmm. Okay, thank you very much.
Thank you.
Any other question for Mauro?